Okay. Hey, everybody. How are you? My name is Brent, and I wanted to just quickly go over a pretty complicated process in anatomy and physiology that a lot of my students have problems with. And this is all about how certain hormones work. How do they trigger, trigger a cell to do something? And that's basically what it's all about. We have some hormone that's been produced by another gland. It's going to activate some cell, it's going to bind to some cell, and it's going to make that cell change what it's doing, either on the inside or the outside. Okay, so how does all that work? And really, very importantly, what are the important terms that we have to keep in mind? Now, I'm going to focus on one type of this signaling and triggering. There are actually two different types. So let's start off with, if we go back to general biology, if you took general biology, you should have talked in one chapter about something called cell signaling. And that's all that hormone binding and activating a cell is. It's just a type of cell signaling. And the same can be said of when a nerve activates a muscle to contract. So we'll see that both nerve cells and hormones, glands, work through this cell signaling pathway to make a target cell do something that that target cell should be doing. Let's see what we got. Let's go over that. Right? Okay. Cell signaling. And, we can say this is how some hormones work. Now that's important because there's a whole different class of hormones I'm going to talk about later that work differently from this. So this is a specific type. And which ones? These are your hydrophilic hormones, proteins, things like that, that bind to surface receptors on the outside of the cell. Okay, so how some hormones work. And again, we'll come back to the other class later, which is different. Just so you know, we also have lipid-based hormones, steroid-based hormones that cross the cell membrane and go in and they mess around with the genes. They turn genes on and off, and in that way affect what the cell is doing. Okay, but let's focus on this today, cell signaling. Now, an overview real quick, how this is working. I've got a blood vessel running, and out here in the tissues, I've got some cell, and it has a very specific type of receptor on it. Somewhere upstream in the blood, there's some gland that has produced hormones. The hormones are, in the blood in a certain concentration, coming through the capillary bed, all right? Now, when I do these lectures, I'm going to remind you of the basic principles from cell biology that you might have gotten before if you took the course. How do these hormones know to come out of the blood? Well, they do it. They're driven out of the blood by a process called diffusion. We have a high concentration here. We have a low concentration out here in the extracellular fluid. So some of these guys are just going to come out down the concentration gradient, right? So they're going from areas of high concentration to areas of low concentration. Now, I didn't draw these properly here. I didn't want to take the time. But let's say all these hormones are actually little box-shaped molecules. They have a specific shape, and that shape needs to bind to a specific receptor. Now, if you look at these two receptors, we see that they have a specific shape to their binding ends out here. And you can look at this and you can say, okay, well, which one is this going to trigger? Because these might end up having very different outcomes in the cell. And if I've produced this hormone, I want to have this outcome. I don't want to trigger this one. And the way that's done is a process called, and this is an idea that, again, also something very important from Gen Bio that you might have gotten, receptor ligand binding. So we've got one diffusion of the molecule from the blood into the ECF, right, the extracellular fluid, bathing these cells. Now, next we have receptor ligand binding. So this is the receptor, and it's going to bind to the ligand. The ligand is another name for the thing that binds to the receptor. So like I said before, let's say these are box shaped. Then what should happen is that hormone should bind to the specific receptor. Right? So there's specificity. I don't turn on things I don't want to turn on, but I do turn on things that I do want to turn on. Now, how do you get a signal here from outside the cell to the inside of the cell where you want things to happen? Remember, all the biochemistry, most of the biochemistry is going on in here, and we need to change the metabolism, the action of the cells. And we'll come to what that means in a minute. So this receptor binds to the ligand outside the cell. And the next thing we need is a process of signal transduction. So don't have much room here. So let's see the third thing. Signal transduction. And that means it's transducing the signal from this side of the cell, the outside of the cell, down to the inside. Now, the way this works is when this binds, you kind of cause some tickling action over here. The protein is misshapen a little bit. It's goes through what they call a conformational change. And this causes something on the other side to become active. And typically what we have is on this side, let's just put a big G, because this is, I'm sure you can see that now. Zoom in a little bit. We've got this G because you've probably heard in your class of the G protein, right? G protein coupled receptors. Now, once this ligand binds to the receptor in a very specific fashion, it activates this G protein. Now, what is this G protein going to do? It's going to activate more things downstream. Now, once we get the signal transduction here, once the signal has been transduced, these are all very important terms. They're all certainly terms that your teacher is going to use in their questions, and they may even directly ask you what they mean. But certainly, as you're reading through a question that talks about G protein coupled receptors or receptors or transduction, you should know what this means. So make sure you pay attention to the terminology so that you recognize it on your tests. Now, the G protein, what it does, it wants to activate the production of a second messenger. So where are we? Number 4. Right? We've got a second messenger. Now, typically, I think, mostly in A&P, the one they're going to talk about the most is something called cyclic AMP. So the cyclic AMP is going to be produced after the G protein. So this takes, and we start making cyclic AMP. Now, I'm not going into the details too much about which enzymes are going to do this because it'll just complicate things too much right now. The first thing we want to get is just a basic understanding of how this works. And then, you're going to have to go in and you're going to have to say, is this an adrenergic receptor? What is it going to respond to? Like, what is exactly the second messenger? What is the enzyme that activates the second messenger? Because it's not the G protein. The G protein is actually activating another enzyme that's going to activate cyclic AMP or make cyclic AMP. Now, cyclic AMP, in turn, is going to kick off something called a signal cascade. If you think about a cascade, if you've been to a river, a creek in mountainous areas, you'll see often that the river runs down and it'll just cascade over the rocks. So it just leads to next steps, right? That's all this means. We've started with a signal, we've got the first messenger, the external messenger. Then we go to the second messenger, the cyclic AMP, and the cyclic AMP kicks off a signal cascade. Now, this is important. What does that mean? Once again, if I've got a series of reactions in a pathway, in a metabolic pathway, then I start at the beginning and it'll kick off the following reactions, biochemical reactions. So let's just do easy here. Let's say we've got A plus B goes to C. Now I've made some product. Of course, what this means is I've activated the enzyme that catalyzes this reaction. And the reason this enzyme is activated is because of the second messenger. Now, I've got my C in a pathway. What is it? A series of biochemical reactions in which the product of one reaction is the substrate in the next. So I take my C, it's the product on this side. This is my arrow pointing this way. So then I take C plus D yields E. All right, now my product in the first reaction has become the substrate in the second. Right? Now I've got my product here, E, and of course, in the third reaction, I'm going to take E plus EF yields G. So I've got this signal cascade. We're going down, down, down the chain, activating things, making things along the way. That's all that is. Now, that's my signal cascade. Along with the signal cascade, we get something called, very importantly, you should understand what this means, signal amplification. So the process of signal amplification means if I have one hormone binding to one receptor, then I'm going to activate one G protein. And that G protein is going to activate your adenylyl cyclase, which is going to make cyclic AMP. But this one active enzyme here might make 10,000 copies of an enzyme. Right? So the next thing is each of those 10,000 activates 10,000 enzymes. So we've gone from having 10,000 activants from 1 to 10,000. Then we go from 10,000 to 10,000 times 10,000. That's 100,000, right? Then what do? The next step, we've got 100,000 enzymes that each activate 10,000. So now we're up to what? 10 million, a million, right? I'm sorry. So we've gone from very weak signal to a very strong signal because at each step, each enzyme is resulting in maybe 10,000 products that then activate 10,000 products, that then activate 10,000 products. So at 10,000 times 10,000 times 10,000, the signal can get really strong inside, and thus the signal has been amplified. All right, what is next? Okay, before we move to the next thing, let's talk about, just for ease of recognition and how to think about things. There's a couple enzymes that play a very important role in this signal cascade, signal amplification, and you'll see these terms. I'm going to explain this in a different video, but kinase is a term you should probably recognize. You're going to see a lot of kinases. This kine comes from the word kinetic, which means action. So very often these kinases are activating some enzyme. Like I said, if I have A plus B here, I can't do anything until I have my enzyme. So I've got some inactive enzyme bouncing around, but once I activate my kinase, it activates this enzyme and I can make my product. So the term kinase that you're going to see in all these signal cascades, these signal transduction pathways, comes down to, it's an enzyme. Once again, remember what ACE at the end of a word means. Whenever you see ASE at the end of the word, that's telling you it is an enzyme. It's going to catalyze some biochemical reaction. Okay. In the case of kinases, these are enzymes that activate something, right? Protein kinases, whatever. Now, the other word you'll see a lot of is phosphorylase. Okay, what does a phosphorylase do? Well, what is it? Once again, it's an enzyme. You see the ending, ACE here, and it's going to perform some action. What is the action? It is to phosphorylate or put a phosphate group onto something. Why is that important? Well, activating and inactivating enzymes is often accomplished by adding a phosphate or taking one off. Now, what's the other thing we can do? We have phosphatases that can also take off phosphates. Okay, so as you're looking through these signal cascades, when you're inside the cell, you're probably going to see some kinases, you're probably going to see some phosphorylases, phosphatases, and that's just telling you what these guys are doing. We're at 6 or 7 now. Don't remember. I'm going to go to 6. So finally, the last thing you're going to do is activate some specific enzyme to do a specific job. There's a reason this hormone was produced. Let's say your blood sugar's low and you have, well, let's not say blood sugar's low, let's do a different one. Let's say you've just seen a lion leaping out from behind a bush and you're in danger for your life. You're probably going to start producing some adrenaline, also known as epinephrine. The adrenaline has to do certain things to your body. It needs to get your heart pumping. It needs to get your blood vessels constricting and dilating in certain places. You want blood to flow to your skeletal muscles, not to your digestive system. So you're going to shunt blood away from the digestive system toward the skeletal muscles. This is what the fight or flight response. If you've gotten to the nervous system, you may have gotten to that yet. Fight or flight in response to perceived dangers triggers this response. And the end result is you need to fight or flight. Now, you're going to breathe, airways are going to open up, you're going to breathe faster, your heart's going to beat faster, blood's going to go to the right places. But another very important thing you're going to need is energy for your muscles, right? So epinephrine or adrenaline is going to bind to this beta adrenergic receptor. It's going to activate a G protein. It's going to kick off the production of cyclic AMP, which in turn turns on a signal cascade through those kinases and phosphatases, and we're going to get signal amplification as we go down the chain. And finally, we're going to get a ton of these activated enzymes. Now, what is this enzyme in the case I'm talking about? So here's an example of what you might end up seeing, and that is the end outcome could be glycogen olysis. Why do we want to perform the process of glycogen olysis? Well, what is it, first of all? It's lysine breaking down glycogen. Okay, great. What is glycogen? Glycogen is a complex carbohydrate that generates, and you'll see this term a lot in your science, gen. Whenever you see gen, you might have seen the word fibrinogen. It's the precursor to fibrin. So when it's cleaved and it's activated, it generates fibrinogen. So that's a term that you can get used to seeing. Now, this is the process of breaking down this complex starch in order to generate glucose. So if I've got my long chain glycogen and I want glucose, what I need to do is pop these little guys off, hydrolyze these bonds, and give you, this is not the same as this, right? Ignore this, this is hormone. This is just a simple example of a glucose molecule, and this is your chain of glycogen. The process of glycogenolysis is going to require that we have some enzyme to come in and pop these off. Now I have individual glucoses, and they can go into the blood. and be carried to the muscles. So that's the process of, cell signaling for these hydrophilic molecules. They bind to surface receptors. They tickle the receptor. They bind very specifically to surface receptors. They tickle the G protein. The G protein activates an enzyme that makes perhaps cyclic AMP. Cyclic AMP, then the second messenger, that's signal transduction. This is the second messenger. Cyclic AMP kicks off a signal cascade where we're activating a pathway of reactions. This pathway, that signal cascade, results in signal amplification where we get a very high signal from a very low signal. And finally, we get this end result, the outcome, the whole point of having produced this hormone, in this case, would have been to make sugars out of glycogen. So to break down glycogen into glucose so it can go to the muscles that are going to need, you're going to need the energy to fight this lion or run from the lion. Okay, I hope that helped. Would appreciate any feedback, and I may continue doing these videos like